I want to quote from a guy, you may know him, you may not be familiar with him, but this is a guy by the name of Professor Fritz Albert Popp.
Yep.
And I suspect that that's going to become a fairly important name in your studies of biophotons. I'm planning on having Professor Popp do an entire series and course on biophotons, but this is what he said, and I quote, "We know today that man essentially is a being of light," end quote. We know today that man essentially is a being of light, and from the photobiology standpoint, we are literally light solidified by gravity and electromagnetic forces. And as far as healing is concerned, the implications are, they're pretty enormous. And if you've been around the BX community for a while, you probably are going to recall that the BX energy catalyst, at least, if we're going back a few years, has often been described as a photocatalytic oxidizer. Anybody ever hear that phrase? I think I kind of want to hone in a little bit into a discussion about light as a prominent feature of cellular metabolism. That's probably will end up being the focus of the lecture for the most part. But returning to Professor Popp, I want to quote him again. He says, "We are still on the threshold of fully understanding the complex relationship between light and life. But we can now say emphatically that the function of our entire metabolism is dependent on light." So if you think about the significance of the BX, particularly the energy catalyst as a molecule that exploits the potential of restoring mitochondrial function, restoring metabolism, think about that statement. You can say emphatically that the function of our entire metabolism is dependent on light. And Professor Popp's, the guy's published 8 books, I think around probably 150 scientific journal articles and different studies on the role of biophotons in human health. We're going to get some of those resources. I'm not sure I can't remember how many of them are translated into English, but we're going to get a lot of that stuff into our library. This field of I said that we reference the field as photobiology, and I think Pops talks about it as being what he calls integrative biophysics. That's going to be a major focus of Delta's educational endeavors. As we move forward, you're going to see integrative biophysics as a very, very important part of what we're all about. What Pops has discovered is that, and this is going back to the 1970s, really, where he discovered that biophotons are involved in virtually all biological processes. And in fact, the original to discover the biophoton really is a Russian going back to like the 1940s. But so if we ask what biophotons are, which might be a good place to start, you could imagine them as a particle of light that doesn't have any mass at all. And what Pops has discovered is that biophotons are, of course, involved in metabolism, that they're really involved in virtually all biological processes. So you talk about light not having any mass. Biophotons are photons, but they're just a little bit different. They're photons of light. They're in the ultraviolet spectrum, but we're talking about low, very, very low visible light range. They're produced by biological systems. Can you see them? Yeah, you can see them, but probably not with the naked eye. They're detectable above what we would say the background thermal radiation, above the background of thermal radiation. They're emitted by tissues. They're emitted by, we can see them in cell cultures. In fact, I have right here in my office a little device that we use that hooks up to a microscope. It's called a PTI fluorescent controller, where we can actually see biophotons. There are a lot of different little cameras, CCD cameras and things that you can use, but they're detectable, emitted by tissues, and they're emitted at normal temperature, which is really something quite interesting about biophotons. We can measure a biophoton using something like a photo multiplier. We can measure them with a special camera like the one I have here, which is called a CCD. They're what we call, if you think about biophotons, they're ultra weak photon emissions, ultra weak. And their intensity coming from the body is probably about 1,000 times lower than the sensitivity of the naked eye. Numerous aspects of biophoton emission from living systems that we should discuss. Professor Pops, of course, refers to them as biophotons as being universal, kind of a universal mediator of all biochemical processes within the cell, which is really amazing considering that every cell generates about 100,000 chemical reactions per minute. And to this day, scientists really can't even explain how, this many reactions, most of which are totally codependent, take place at exactly the right moment, exactly the right place, 100,000 reactions a minute, and everything just seems to work in perfect harmony. So, if you look at Professor Pops, he and his colleagues have been able to demonstrate that every single molecule requires precise photon excitation in order to generate a chemical response. In order for photons to, because if you look at a photon, it's really a facilitator, but in order for them to function in that capacity as a facilitator, they really have to possess an extraordinarily high degree of coherence, and they do. And when you compare photon signaling to say something like an electromagnetic field, and we discussed that on the last call about field potential within the cell. When you compare photon signaling to an electromagnetic field, let's say it's a thermal response, which is electromagnetic, which is very common throughout the entire biological landscape, you have infinitely greater coherence and order with photon signaling than you do with the electromagnetic field, which is very disordered. So from Pop's perspective, photons are kind of the source of all signaling patterns throughout the entire biological terrain. And I think you can find that, if you look into the literature, you're going to see a lot of stuff on photon modulation, biophoton modulation. It started with Pops, but now it's prevalent throughout the literature. The primary transmitters of information governing every single reaction within the system, we're talking about information from cell to cell, information within the architecture of the DNA, within the cell, between the various systems, between cells, all modulated by biophotons, which makes a lot of sense considering how efficient biophotons really are. They can trigger responses in a way that they generate essentially no heat, and this is one of the reasons why they're so interesting to study, much different than something that's based on electromagnetism. There's lots of heat going on there, which could be seen as interference. If you're, think about the analogy of two radio towers, or a better analogy would be the fact that my phone call had all that static on it. That would be kind of like thermal interference. So the communication is really impaired in that manner. But photons at the speed of light generate incredibly clear signals. We previously discussed the energy catalyst, and if you've listened to any of the lectures, hopefully you've gone back and listened to the lecture where I discuss it from a couple different perspectives. I discuss it from the biochemical and the quantum perspective, but you may have asked yourself why the BX energy catalyst is one of the things that's kind of interesting is the catalytic aspect of it. How is it capable of generating such widespread production with so little of the solution that's actually there? I think I mentioned last week to you, shared some numbers with you on a case that's currently at the TC. One spray generating an 82% increase in hydroperoxides that we measured via spectrophotometry. That was just, I think, last week at the training center. A lady visiting from the UK, 82% increase, one spray. So how in the heck, is that possible? This was and that was a 12x preparation. I mean, that's one part of the molecule to a trillion parts of water, and we had an 82% increase in hydroperoxides. Well, if you're wondering how that's possible, I'm about to tell you. And from the perspective of the biophoton as the principal modulator of that catalytic process, I don't think I've ever really discussed that before. But the toxin, think about the toxin, usually in the form of an unsaturated protein. These are toxins that are secreted by stealth pathogens. It can be a carcinogen. It can be a virus. It can be an amine group. There are a few different possibilities, but let's just stick with the toxic protein, if you will. That toxin is dehydrogenated. I think everybody understands that. We've discussed that in great detail in the lectures, but how that happens, the mechanism of the BX is described or has been described as a photocatalytic process. But what happens, and I did get into the catalytic aspect, but the electrons from the BX essentially are migrating over to the toxin. And sometime at the point of dehydrogenation of the toxin takes up a photon, okay? And we can measure this with a counting module. What's amazing is that if you just add, let's say, hydrogen peroxide to living tissue, usually you'll see an immediate increase in the emissions of photons, not in the absorption, but in the emission of photons. Take a cell culture, put H2O2 in it, you're going to see an increase in the emissions. But what we actually see is a decrease in the emissions, which is a good thing because we're being able to measure that as increased absorption of the photon, suggesting that the BX is somehow stimulating photon absorption while generating peroxide peaks needed to properly regulate the cell. And because the biophoton is really, really, really efficient, meaning again that it doesn't generate a lot of heat, it's immediately available to sustain the cascading of peroxide formation. Okay, so in a sense, the BX stimulates the uptake of light. I want you to understand that. It's pretty important. If you're going to really understand why it's catalytic, you need to understand the role of the biophoton in the process. The BX stimulates the absorption of biophotons, and that increased photon absorption is directly correlated to peroxide formation, and we've measured that. You could say that the BX catalyzes the biophotonic conversion of either water. So if you look inside the toxin, what we're really talking about is a conversion of water plus singlet oxygen to hydrogen peroxide, or hydrogen, this straight up hydrogen combined with molecular oxygen to form a peroxide at a rate of about 30 molecules of H2O2 per photon. The wavelength of the photon is, I don't know if I've ever discussed that either, but it's around 312 nanometers, which is just under the visible range, but still in the UV spectrum. It's just under the visible range. This is why we can optimize the production of H2O2 with the use of certain photodynamic therapies within that spectral range. We can stimulate this process in the absence of UV and get no production of peroxides at all. So consider the significance of light in the production of H2O2, and consider the importance, obviously, of H2O2 as a signaling agent in general. You think about H2O2 in human health, if we're just talking about as a general, from a general perspective, how important is it in cellular function? We're talking host defense, we're talking controls, it controls really almost all of the biochemical pathways involved in regulating redox signaling pathways, regulates transcription factors, regulates cell death. It's involved in pathogen protection, immune modulation, a bunch of different stuff. And so, but more specifically, the role of H2O2 with the BX is the production of a peroxide free radical. I'm going to test you within or without the toxic structure. Within, right. You remember the analogy that I use about the building kind of imploding, the difference between strategically placing those explosives on the structure of the building as opposed to trying to light them off 3 blocks down the road. Internal production of peroxides within the toxic structure. In review that fact, if you need to go back to the videos, but that's really a huge part of the BX novelty, lies in its ability to generate the free radical internal to the toxic structure, much different than something like peroxide therapy is an IV therapy, or anything that's an external application. And so let me emphasize this again, this very important part. The BX energy catalyst catalyzes the biophotonic conversion of hydrogen peroxide within the toxic architecture. Okay, BX catalyzes the biophotonic conversion of the peroxide within the toxic architecture. Now, I hope you realize that by advocating the idea, the ludicrous concept that light emitted from our cells carries information, is, you guys know that's a heresy, right? From the viewpoint of of the mainstream thought. This is just not what they teach. It's not what they believe about the inner workings of the biology. Coherent energy from this light, from the biophoton, could organize and influence matter at every level of the biology. And so you'd be called absolutely crazy if you believe that. And hey, I'm crazy. In fact, I come with a very long line, I come from a very long line of lunatics. And in fact, I attract lunatics, as evidenced by the people that are on this call right now. So, back to the question that we started with, what are you? You are a highly structured and organized frequency. And you're synchronized. You are a synchronized, coherent bundle of light, literally. And you're going to have to explore that as we build out that section, and we have the emphasis on integrative photobiology. But I want to quote from a guy by the name of Dr. Dispenza. He's kind of an enlightened researcher, kind of a kindred spirit on the subject. He says, and I've talked about Peter, who's a friend of mine before, a Russian scientist named Peter Garyev, I'm quoting here, placed DNA inside a quartz container and zapped it with a laser. Now, of course, this was a 727 nanometer laser. In fact, I've done this exact experiment, but he zapped it with a laser, a very coherent form of light. The DNA absorbed the light and stored it for up to 30 days inside a corkscrew-shaped spiral in an exact blueprint of light reflecting the DNA pattern. Even more interesting is that the spiral of light stayed in the same place even after the quartz container and the DNA had been removed. You guys following that? Kind of almost like an echo, a biophoton echo. Yeah. He goes on to say that Garyev's work suggests some unknown force is holding the light in place. One possible explanation is that the DNA is responding to an external energy field. This energy field is exchanging information with your cells in the form of light. In essence, our bodies are working as a giant antenna that is constantly sending and receiving signals from the field. And of course, that might remind you a little bit of our discussion about George Lakhovsky and his theory of really kind of postulating the existence of the delta helix as an emitter that received and broadcasted frequencies with the very short wavelengths. And this is the reason why I developed the fourth phase water system with the ability to create an exclusion zone and then infuse light of a specific wavelength into that zone of the water in a manner that the photons combine and form a light molecule. That's something that I'll probably have to get into on a lecture on its own, but This is actually a technology that I've developed, and there may be some on here that have used that. But so imagine drinking liquid light to kind of enhance the availability of the biophoton. There's a lot that we can do there with photodynamic stuff. There's several ways that we can enhance that process, and this will be a pretty big part of the equation. The photon obviously the biophoton is a huge part of metabolism. It's involved in every single aspect of really kind of every reaction that occurs with all of the reactive oxygen species, including hydroxyl radicals and superoxide, is all dependent upon the uptake of the biophoton as the modulating force. And you can go type this into the literature if you think it's really crazy. I mean, once you start getting into the literature, it's in the mainstream now. They're starting to really get deep into this. Of course, most of the people we're studying are so far ahead of their profession that right now they're just starting to have the discussion that Professor Pops was having back in the 70s. But you'll find it. I mean, I found 3 NIH studies, just reviewing it last week. 3 recent NIH studies on the role of the biophoton. So it's starting to be accepted as a modulating force, but whether that gets translated into anything useful coming out of the mainstream is not likely. So if you guys want to listen to an inspiring song, there's one that's called, it's by a kid by the name of David Archuleta, who was on American Idol years ago. You may remember him, but he sings a song called Glorious. And the lyrics speak of life as being this glorious symphony. And Professor Pops actually talks about this concept. You think about the Newtonian model, one reaction leads to another reaction that leads to another reaction that leads to another reaction. It's a very rigid kind of construct. You contrast that with the role of biophotons in human health, and what you'll find is that our biology does in fact behave much more like a symphony than it does like this rigid biochemical process that's described by the mainstream. Just remember, you're a light being, so don't let anybody give you any crap. Your cells, the bottom line here is, you think about the implications, your cells emit light in really everything within the cell. Every membrane, every constituent, every cellular component, including the DNA, emits light in the form of a biophoton. And that light is constantly being emitted, and it's being received in the form of information, which is really amazing to think about. And if we can just learn how to control that light, or at least facilitate light modulation, then we have the ability to change ourselves at probably the most fundamental level of the human experience.